Okay, so I thought I would go ahead and do some of these genetics problems with you, or actually all of the genetics problems with you, just so that you can check over your work and make sure that you understand how each of these problems is solved before you go ahead and you take any quizzes or exams on this material. So I'm going to start off right with question number 1. And again, this sheet of genetics problems, part 1 questions, is right on Blackboard for you. So you want to make sure that you download that. You're going to have to work through these problems, so it's best to download it and write, and then take pictures, create a PDF or a Word doc from including your pictures, and then upload that to Blackboard. Okay. If you want to type it, that's fine as well. It's just that you're going to have to draw some Punnett squares, and that might be a little tricky for you. So let's start right with number 1. So number 1 says, in squash, a gene for white color, big W, is dominant over its allele for yellow color. Okay, so we're dealing with squash, and the gene is color. Okay, so again, I like to write everything out, and I'm going to write with my finger here, so I'm sorry if it's a little bit messy. So white is going to be the big W allele, and yellow is going to equal the little w allele. So I'm going to draw a line over that little w to make sure that we know that it's lowercase. It says to give the phenotypic ratios for the results of each of the following crosses. Okay, so the first cross is big W, so homozygous dominant white squash being crossed with little w. So again, homozygous recessive yellow squash. And hopefully you know, just by looking at this, that all of the offspring would be heterozygotes, big W, little w. Okay, and what color would they be? White. So you would say 100% white, because it asks for the phenotypic ratio. Okay, let's say you didn't know that off the bat. Let's look at this parent first. Okay, so think about meiosis. Okay, think about gamete formation, and hopefully you can see that all that this parent has is the big W allele. And so 100% of this parent's gametes are going to be big W. The same thing is true for this parent. It's little w, and so all it has to donate, to give, is going to be the little w. So our Punnett square would look like this, really simple. And inside of there we would have big W, little w. Okay, so again, 100% white heterozygotes. Okay, so that's that first part. Let me get my eraser, and we're going to erase just some of this. We'll go to our next cross. Okay, and grab my pen. Okay, so our next cross says big W, little w, so we have a heterozygote. Try to use that terminology as much as possible. And we're going to cross that individual with a homozygous recessive yellow squash. Okay, so let's make our Punnett square. So we'll start with this parent. Okay, so half of this parent's gametes are going to contain the big W allele, coding for white, and half of this parent's gametes are going to contain the little w, the yellow allele. Okay, let's go over here to this parent. All this parent has is the little w allele. So all of the gametes produced in that parent are going to be little w. So we can just do 1. Sorry about that. That was my timer. Okay, so now let's fill in the inside of the box. Remember, the inside of the box shows the potential offspring. So in my first box, I'm going to put big W, little w. Okay, so again, that's if this gamete is fertilized by this gamete. Okay, in this box, the second box here, it's going to be this gamete with this gamete. So it's going to be little w, little w. And so here, you would end up with a 1-to-1 phenotypic ratio. 1 heterozygous white squash and 1 homozygous recessive yellow squash. Okay, let's do that last one together. A little slow at erasing. Okay, so let me grab my pen again. So we have 2 heterozygote white squash. We're going to cross those. And I hope from lecture and maybe some work in lab that you realize that anytime you cross two heterozygotes, you end up with a 3-to-1 ratio. So that should pop up in your mind real quick, 3-to-1 ratio. So let's do this. Make our Punnett square. So let's do this parent first. We're going to put its gametes along the left-hand side of the box. So half of this parent's gametes are going to contain the big W, the dominant allele, and half will contain the little w. And the same thing with our other parent. Half will be big W, and half will be little w. So there's the beginning of our Punnett square. Remember, across the side and the top, we represent gametes from each parent, and inside the boxes are potential offspring. So if this gamete is fertilized by this gamete, we end up with big W, okay, or homozygous dominant white. Second box here would be this, with this, big W, little w, heterozygous, still white. Here, big W, little w. Again, heterozygous, but still white. And in our last box, we're going to have little w, homozygous recessive, and that's going to be our yellow. So hopefully you can see, we'll grab a different color here, where we have our 3-to-1 ratio. So for every yellow, there should be 3 white offspring produced. Okay, so that's it for question number 1. Let's go to question number 2. Okay, so question number 2 says, if pollen from a heterozygous white-fruited squash plant, okay, so we're dealing with the same gene, the color gene in squash, and so it's still going to be big W equals white, and little w equals yellow. Okay. So we've got pollen from a heterozygous white-fruited squash plant. Okay, so big W, little w, heterozygous, white. Okay. We take pollen from that plant and we place it on the female part of a true breeding white-fruited plant. Okay. True breeding means homozygous. And it's white-fruited, so it must be homozygous dominant. So that means we're going to cross that with big W. Okay, this here is the female, and this here is the male. Okay, so we said pollen was placed on the female part, so there you go. So it says, show using ratios the genotypes and phenotypes you would expect the seeds from this cross to produce. So you're just going to do a simple Punnett square. So again, this parent, I'm going to put here, half of its gametes are big W, half are little w. I'm going to go ahead and put this parent across the top. All of its gametes are big W. Inside the box, I'm going to put potential offspring. I'm going to grab another different color. Okay, so this first box will be big W, and the second box is going to be big W, little w. So, for a phenotypic ratio here, you would say 100% of the offspring will be white-fruited plants. Now, a genotypic ratio would be 1-to-1. Okay, 1-to-1, homozygous dominant to heterozygous. Okay? So you always want to make sure with these genetics problems that you actually give the answer, right? Just putting this Punnett square is not enough. You need to actually answer the question. So it says using ratios, the genotypes and phenotypes. So you would say 100% white, and then you would do a 1-to-1 ratio of of homozygous dominant. I don't have room to write it. So you do a 1-to-1 ratio of homozygous dominant to heterozygous. Okay? And when you do these genetics problems, wherever your answer is, what you should do is you should box it in. Let me grab my pen. You should really box that in to show me that that's your answer. Okay, that'll help me as I'm grading these to not have to look through every single part of the problem. I can just look at your answer, and if it's correct, I can go to the next problem. If it's incorrect, I can look back at your work and try to find some credit for you. Let's go on to the next one. Okay, so number 3 says, in human beings, brown eyes are dominant over blue eyes. Suppose a blue-eyed man, oh, hold on, let's do this. So we were talking about eye color, and we're going to say brown and blue. Those are the two phenotypes. Okay, it's really pigmented and not pigmented. So brown is going to be dominant, so I'm going to use a capital letter E. I'm going to try to do the same that we did in lab. And blue will be a lowercase letter e. Okay, so now it says, suppose a blue-eyed man whose mother had brown eyes. It doesn't really matter what his mother had. He is little e, little e. This is the man. He's got blue eyes. He's going to mate with a blue-eyed woman. So she's also going to be little e, little e. So woman. Okay. They have 4 children. 3 have blue eyes. So 3 have blue eyes. And 1 equals brown. It says show how this is possible. Well, this is a trick question. It's not possible, right? Because neither mom or dad carry the dominant big E allele. Okay, so it's not possible. Hopefully you didn't get too hung up on that one. Trick question. So that would be your answer. You would say this is not possible. Box that right in. Number 4. If a brown-eyed man, okay, so same thing, eye color. So here's man. He's got brown eyes. Okay, so I know that he's big E, and I'm going to put a dash to represent that I'm not sure yet what that second allele is. Okay. The next part of this question says whose mother had blue eyes. So if his mom was little e, then he must be big E, little e, because his mom only had the little e allele to give. Okay? His dad could have been big E, or big E, little e, and his dad would have had to have brown eyes. Okay, but he definitely inherited that little e allele from his mom because his mom has blue eyes. Okay, he mates with, or marries as I say, a blue-eyed woman. Okay, so here's the woman, or the mom. Okay. They have 10 children. And all of them have blue eyes. Can you be certain of the man's genotype? Yeah, absolutely, right? He must be this. Number 1, because his mother had blue eyes, but number 2, because his children have blue eyes. So he must have that recessive allele. He's definitely a heterozygote. So again, you put for your answer, yes, man is heterozygous, big E, little e, and box that right in. Okay, let's erase that. So we finished questions 1 through 4. Let's go to question number 5. Again, it's about eye color. So it starts off and it says, a brown-eyed man. Okay, so we've got man. Ooh, I gotta grab my pen. Sorry, hold on. Pen. Okay, so we got man, and he's brown-eyed. And we don't know what that second allele is, right? Whenever there's a dominant phenotype, we're not 100% sure on the genotype to start. We might be given other clues that are going to tell us what that is, but right now we don't know. So we've got a brown-eyed man whose father was brown-eyed and whose mother was blue-eyed. Okay, now since mom had blue eyes, we know the second allele is the little e, right? So mom is little e, dad is, well, we don't know, big E dash. He could have been big E, or big E, little e. We don't have enough information, but we know that the man here is definitely big E, little e, because mom only has the little e allele to give. Okay, that's all she has for gametes. Okay, so he's going to mate with a woman who has blue eyes. Okay. They have a blue-eyed son. Notice how I like to write every step of the problem. It just helps you to keep track. Word problems are tricky, so keeping track of what you're doing keeps you organized, and you're more likely to come to the right answer. Okay, so then it says, for which of the individuals mentioned can you be sure of their genotypes? So again, you would put man, big E, little e, woman, little e, son, little e. And then from the man, you can draw off and you would say his mom and dad, or you can put grandma and grandpa, however you want to do it. We know grandma is little e. Grandpa is big E dash. So that's the only one that we don't know for 100% sure what his genotype is. And the last part of the question says, what genotypes are possible for the others? And so right here, Papa could be big E, or big E, little e. We don't have enough information to figure out what his genotype is. That's it for number 5. Okay, let's clear this. Now it's going to get a little bit trickier. So everything we've done up until now has been the monohybrid cross, right? So mono meaning one gene. We follow just one gene at a time. Maybe it was eye color, or maybe it was fruit color and squash. But again, we were just looking at the inheritance of one single characteristic. Now, questions 6 through, 8 or 9 here, 9 I think, we are going to be doing the dihybrid cross. Okay, so in these examples, we're going to be following 2 traits at the same time. Okay, a little bit more difficult. So question number 6 says, in peas, a gene for tall plants is dominant over the allele for short plants. Okay, so I want to keep track, right? So I'm going to put tall. Oh, I lost my pen. Did I? Okay. Tall equals, I'm going to use big T. And short equals little t. Okay, there's one gene. And then we've got shape. So the gene for smooth peas is dominant over the gene for wrinkled peas. So smooth equals big S. And then wrinkled equals little s. Okay, so I've got 2 traits. I've got the height gene and the shape gene. Tall is dominant to short, and smooth is dominant to wrinkled. So I've got all my alleles present there, right? Okay, so then it says calculate phenotypic ratios for the results of each of the following crosses. Okay, so our first cross, we've got an organism that is going to be big T, little t, big S, little s, so we could say heterozygous for both genes, and we're going to mate that organism with an organism that has the same genotype. So again, heterozygous for both genes. So both of these individuals would be tall and smooth, okay, but again, they're both heterozygous for both traits. Now, you might know just by looking at this that this is going to give you that 9:3 to 3:1 ratio, okay? Hopefully you just kind of know it like that. You're going to see it, we saw it in lecture, you're going to see it in lab. So hopefully you know that that's what this type of a cross would give you. But just in case you don't know that yet, let's go ahead and do the Punnett square. This is going to be a big one, right? It's going to be that 16-box Punnett square. And remember, when we set this up, let me grab a different color, we have to show all potential gametes, okay? All potential combinations of alleles in gametes, okay? Now, these genes are not linked. They follow Mendelian rules, and so this is how it's going to be. So let's start off here. We could have the big T allele in this parent end up with the big S allele, okay? So again, you have to remember back to meiosis. Okay, that's one possible genetic combination in a gamete. Or we could have some gametes that are going to be big T, little s. Equally as likely. And then it's equally as likely that gametes that are little t, big S. And equally likely of little t, little s. Okay, so there's 4 potential gametes, and each are equally as likely to form. So we say 1/4 of this organism's gametes will be each of these genotypes. Okay, all right, there's parent number 1. Parent number 2, same thing. We're going to put this parent across the top. So again, we're going to go here, big T, big S, here, big T, little s, here, little t, big S, and here, little t, little s. So again, 4 potential gametes. Remember that law of independent assortment? Gives you all these possible combinations. And notice this is a 16-boxed Punnett square, a little trickier. Let me grab a different color to show my offspring on the inside here. So again, I'm going to take and do just what I did with my simple monohybrid. I'm going to show the results of gametes coming together in that box, and that's going to give me a potential offspring. So in this first box, I'm going to do big T, big S, big S. In this box, it's going to be big T, big S, little s, big T, big S, big S, big T, big S, little s. And now I'm going to go over, so I'm doing this gamete with each of these down the row. So my first box here is going to be big T, big S, little s. You always want to make sure that you put one gene before the other. So in this case, I'm putting the T gene before the S gene. And you always want to make sure that you put the dominant before the recessive. Okay, so I would never put little s, big S. I always put the dominant before the recessive if they're both present. Okay, next box, big T, little s, big T, big S, little s, big T, little s, okay, big T, big S, big S, big T, big S, little s. Hopefully I don't make a mistake here. Little t, big S, little t, big S, little s. Okay, last row here, so I'm doing this gamete now. So big T, big S, little s. This is a big T. Okay. Big T, little s, little s. Again, I'm putting that line over the S just because S's look the same, capital and lowercase, so I want to make sure I designate that. In this box, I've got little t, big S, little s. And then in this last box, I have my, I fit it, there you go, homozygous recessive for both. Okay, so let's go through now and figure out who's who. So we're doing a phenotypic ratio. And so I'm going to go ahead with my green pen and I'm going to circle every tall smooth. Okay, so any offspring in this chart here, in this Punnett square, that has a big T and a big S. It doesn't matter what's there for the second allele, right? Because tall is dominant to short and smooth is dominant to wrinkled. So just one dose of that allele is going to give you that phenotype. So this is a tall smooth. This is a tall smooth. Tall smooth, tall smooth, tall smooth. Nope. Tall smooth. Nope. Yep. Tall smooth. Tall smooth. Nope. Nope. Yes. Tall smooth. Oh, hold on. Yes. Tall smooth. Nope. 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 Okay. Let's count them up. 1, 2, 3, 4, 5, 6, 7, 8, 9. I counted. So there's my 9. I have 9 tall, smooth offspring. 9 out of 16. Let's grab a different color here. Let's use red. I'm going to circle all of the tall wrinkled. So big T allele, but for the shape gene, they're going to be little s, little s. So that includes this guy, this one, and this one. So there's 3, or 3 out of 16 are going to be tall wrinkled. Okay, let's use purple. I'm going to circle now my short smooth. So little t, big S, dash. I got 1, 2, 3. So 3 out of 16 are going to have that phenotype. And my last one, I'll circle it with blue. My homozygous recessive there, that's going to be my only short wrinkled. So see that 9 to 3 to 3 to 1 ratio? Again, that's the phenotypic ratio. 9 tall smooth to 3 tall wrinkled to 3 short smooth to 1 short wrinkled. 9 to 3 to 3 to 1. Anytime you cross 2 heterozygotes for both genes, you're going to get that ratio, as long as they follow Mendel's rules. Okay. Next week, in Chapter 12, Part 2, we're going to talk about linked genes, which would not follow this rule. Okay, we'll save that for then. Okay, that was number 6, Part 1. Okay, let's go on to the second part here. Okay, so the genes are still the same. We're going to cross an organism that is big T, little t, big S, little s. So again, heterozygous for both genes. And we're going to cross that individual with a homozygous recessive. Oh, we can call this a test cross, right? Okay, we're crossing with an individual. Well, we know the genotype, so I guess it's not a true test cross. But anytime you cross an organism with a homozygous recessive, you could label that as a test cross. Okay, so I'm going to keep my blue. So, no, let me grab red actually so I can do this. So let's set this up. I'm going to put this parent across the top, okay, just so it lays out a little bit better. So again, I'm going to do this, big T, big S. I'm going to do that, big T, little s. I'm going to do this, little t, big S, and then lastly I'm going to do that. So little t, little s. And then I'm going to go ahead and I'm going to put this parent on the side. And why did I do that? Well, the only gametes that second parent can produce are little t, little s. Oh, I should put lines over these. Because that's all that parent has. So we don't need to write 4 times little t, little s. It's the same thing. Every time you write it, you're going to get the same results. So you can go ahead and you can simplify your Punnett square. Okay? That's the only gamete that parent can produce. Okay, so let me grab my blue and we'll fill it in. So this first box, we're going to put big T, big S, little s. Second box, big T, little s, little s. Little t, big S, little s. And little t, little s, little s. There's that 1-to-1 to 1-to-1 ratio. We did that in lecture too, and you'll see it in lab. So again, 1 to 1 to 1 to 1. So 1 tall, smooth to 1 tall, wrinkled to 1 short, smooth to 1 short, wrinkled. So it's a 1-to-1 to 1-to-1 phenotypic ratio. That one was a little easier. Let's go on to the next one. So we've got little t, big S, little s. And we're going to cross that organism with big T, little s, little s. Okay. So again, we can make some simplified Punnett squares because there's kind of limited gametes that are going to be produced. Because the first gene, the only allele present is a little t, we don't have to take each little t and figure out what it's going to be with. We can kind of do this as a pair. So we could say little t, big S, and little t, little s. Those are the 2 types of gametes that will be formed by this organism. So 50% are big T, big S, and 50% of the gametes are little t, little s. Okay? Let's come to this parent. Same thing. We're going to do big T, little s, little t, little s. Now you could have gone through and did all 4, but you would have had repetition. And so to avoid that, we can kind of simplify things. Okay, so here's my Punnett square. I'll do blue on the inside. So in this first box, this is going to be big T, big S, little s. Little t, big S, little s. Big T, little s, little s. And lastly, little t, little s, little s. If you're uncomfortable not doing a 16-box square, go ahead and do it. But you should hopefully get the same results. And so let's look at our results here. I'm going to grab dark red here. Okay, so this first offspring is going to be tall and smooth, and he's the only one. So 1 tall smooth to 1 tall wrinkled to 1 short smooth to 1 short wrinkled. Okay, so there's that ratio again. Okay, and I think our last one on here is going to be big T, little s, crossed with little t, big S, big S. Okay, so same thing. We don't have to do a 16-box Punnett square because we have repetition of alleles. And so we could say, okay, the T alleles here in the first parent are the same, and so are the S's. So all of the gametes produced are going to be big T, little s. That's the only type of gamete that could be produced. And from this parent, well, we only have little t, big S. Oh, very simple Punnett square. 100% of the offspring are going to be big T, big S, little s. Or 100% will be tall and smooth. Okay? So again, if you're uncomfortable kind of doing it in shorthand, you can go ahead and make a 16-box Punnett square. But again, you don't need to. You can simplify things. Okay, and as you get better at these, hopefully you're able to do that. I'm looking at the next problem, and it is quite wordy. So let's take a look. We've got to make sure we write this one out well. Okay, so we're talking about hogs, and we have a gene that produces a white belt around the animal's body, and that's dominant to no belt or uniform color. Okay, so we've got belt, belted, and you could choose any letter, but I'm going to use a capital B. And that's going to be dominant to no belt or uniform, which is going to be a little b. Okay, and then it says, another independent gene produces fusion of the two hooves on each foot, which is called syndactyly. Okay, so we're going to write syndactyly. Let's see. Yeah. Oh, sorry. My pen. Okay, so I'm just going to put syndactyly. Ooh, sorry, it's my dog. We'll do a capital S there. And then that's going to be dominant for normal, which would be not fused hooves. Okay, so getting through some of that is sometimes hard. Okay, so we've got two genes. One is color, belted or no belt. Okay, do they have a white belt around them or not? And the second has to do with their feet, their hooves. Are they fused or not fused? So syndactyly means fused, and normal would be not fused. Okay. So it says, suppose a uniformed colored hog. Okay, so let's do this. So we've got hog number one. Uniform would be homozygous dominant, recessive. So little b. Homozygous for syndactyly, so that's big S, is mated with a normal-footed hog. So I'm going to put over here, little s, that is homozygous for the belted color, so big B. Okay, this is our first cross. Okay, so those would be the parents. Sorry. And the result of that would be the F1 generation. Okay, now hopefully, just by looking at that, you could see that all of the F1 offspring, oh, hold on one second, let me pause. Sorry about that. So all of the F1 offspring should be heterozygotes. Okay, so how do I know that? Well, let's do the Punnett square. So parent number one, let me grab my pen here. Okay, got it. Parent number one, the only gametes it can produce are little b, big S. And parent number two, the only gametes it can produce are big B, little s. So inside my box here, all of the offspring are going to be big B, big S, little s. So there's the first part of that question. It says, what would the phenotype be of the F1 generation? So you would put, the F1 generation would all be belted and syndactyl. Okay? Or you could say belted fused hooves, or however you want to put that. Okay, and then it says, if the F1 generations are allowed to breed freely, okay, so that would mean this, big B, big S, little s, right? Mating with other offspring that are also big B, big S, little s. It says, what phenotypic ratio would you predict for the F2 generation? And hopefully you know we're crossing two heterozygotes, both heterozygous for both genes. That's going to give you that 9:3:3:1 ratio. And again, if you don't know that yet, go ahead and do your Punnett square. It's going to be that big 16-box Punnett square. You're going to have 9 belted syndactyl, 3 belted normal, 3 no belt syndactyl, and 1 no belt normal. Okay? Okay, let's erase this. Question number 8. So, question number 8 deals with watermelon. So in watermelon, okay, so the first thing we're going to do is a color. So green, oops, do I have it? Is going to be dominant to striped, okay? So I'll use big G for green, and then striped, I'll use little g. Okay, and then it says short shape is dominant to long. So short, we'll do big S, and then long, we'll do little s. Now remember, you can choose any letter to represent those alleles that you want. I just chose G and S here, but you can pick whatever you want. So then it says, suppose a true breeding, okay, when you hear true breeding, you're thinking homozygous. So suppose a true breeding short striped fruited plant. Okay, so short with stripes, true breeding. Let's put the color gene before the length gene. So striped would be little g, and short would be big S, big S. Okay, we're going to mate that individual with a plant that is heterozygous for both characters. So big G, big S, little s. This would be a green short. Okay, it says what phenotypes would this cross produce and in what ratios? Okay, so let's go ahead and put this parent on the left-hand side because it's easier. So this parent only has little g and big S for its alleles, and so all of its gametes are going to be little g, big S. And then my second parent, I have to go ahead and I have to figure out all these, right? So we got big G, big G, big S. And I'm running out of room a little bit on here, so let's do this. Big G, big S, or big G, little s, okay? And then we've got little g, big S. And it's equally likely that little g, little s will also be formed. So I got 4 different potential gametes being formed from that parent. Okay, so now let me grab a different color pen. And this is going to be a little tight here. So first offspring would be big G, big S, little s. So green and short. Second will be big G, little g. Oh, sorry. This should have been big S, big S. Right, big G, big S, big S. Okay, second, big G, big S, little s. See, I'm not with you guys for you to find my mistakes. If you find my mistakes, make sure you do them right on your paper. The next one is little g, big S, big S. And the last one is little g, big S, little s. Okay, so now it says, what phenotypes would this cross produce? So let's see. We'll use dark blue. So this is going to be green and short. This one will also be green and short. Okay, so we've got 2 that are green and short. And then these last 2, let's see if they're different or the same. I'll use purple. This first one is going to be striped and long. And the second one is also, striped and long. So that is a 1-to-1 ratio. So 1 green short to 1, I'm saying that wrong. 1 green short to 1 striped short. I got it right. Okay. So that's going to be that cross. Again, that's going to be your answer. So you're going to box that in. It's a 1-to-1 ratio of green short to striped short. I think I got that right. Okay, last question. Let me erase this. Okay, the last question, number 9. Okay, so we've got the fruit fly, Drosophila melanogaster. Okay, we're looking at 2 genes. One gene deals with wing size, and the other deals with body hair. Okay, we did the same kind of a problem in lecture. So we've got vestigial, which means short wings, and hairy body are going to be recessive. Okay, so that means that normal or long wings is going to be dominant. So I like to use a big V to represent the normal, and for the vestigial or short, I will use a little v. Second part here deals with body hair. So hairless is going to be dominant. Hairless is going to be a capital H, and then hairy is recessive, so it's little h. Okay? Okay, so then it says, suppose a true breeding. So again, when you hear true breeding, you're thinking homozygous. So a true breeding normal winged hairy male. Okay, so normal wings would be big V, and hairy, correct, is going to be little h. We're going to cross that with a male that is with a female, sorry, that is heterozygous for both alleles. So both genes, big V, big H, little h. Okay. It says what types of offspring would be expected. Make sure you show your work and give the phenotypic ratios. Okay. So we're going to go ahead and we're going to take this parent, and we have repetitive alleles here, so two of the same. So the only gametes that can be formed are those that are big V, little h. All of them. No other gametes would be formed. The other parent, a little trickier. We've got to do this, right? Big V, big H. A quarter of them will be big V, little h. A quarter will be little v, big H. I always do this. I don't leave myself room. And a quarter will be little v, little h. Ooh. Okay. Actually, I'm going to rewrite this really quick. Move it over. Okay, I'll do it fast. Okay. And I'll use purple. So again, this parent number 1, is going to be all of its gametes are big V, little h. The other parent across the top, big V, big H, big V, little h, little v, big H, little v, little h. Remember, meiosis gives you an equal likelihood, or a 1/4 chance, that each of those types of gametes will be produced. Okay. Oh, lots of noise happening in my house tonight. Sorry about that. So let's go ahead and put in our potential offspring. So this first box will be big V, big H, little h. Okay, so that offspring will be normal winged and hairless. The next one will be little v, little h, little h. So again, normal but hairy. Big V, big H, little h. Normal, hairless. So these two have the same phenotype so far. And my last one would be big V, little h, little h. Okay, and so these two here and here are both going to be normal, hairy. Okay, so we have a 1-to-1 phenotypic ratio. 50-50, right? So we've got 2 normal, hairless, and 2 out of 4 normal, hairy. So we have a 1-to-1 phenotypic ratio. Okay, so that's it for that first genetics sheet. So again, I want you to check your work. Make sure that you understood how to get these answers. I'm less concerned about you getting the right answer. I really just want to make sure that you know how to do this. So when it comes time for the exam and the quiz, that you get those right. Okay, so hopefully you've watched this video and you've checked your work before you've submitted it.